Yeah, I forgot.
Okay, so we should have some time for review at the end of class. So I want to make sure that we get through this last set of slides before our exam. Although really the part that you should focus on knowing is what we went over on Monday, that kind of summary of the couple of types of glial cells that we want to know exist and we know have functions as part of the nervous system. So what we went through on Monday was the kind of like highlights, sort of cheat sheet version of what we want to know, but we'll talk a little bit more today about the central nervous system just to make sure that we've done it, because next time we're going to move into talking about other parts of the nervous system in our next unit. Okay, so this was the sheet we went over on Monday, talking about our different types of glial cells that have supportive functions for our nervous system. So we have our astrocytes forming our blood-brain barrier, making sure that we have control over what actually comes into contact with our nervous system. We have cells that make cerebrospinal fluid. So hopefully in lab, when you've looked at the brain, you got a chance to look at those ventricles, those hollow areas inside the brain. You also saw the different layers of the meninges that surround the brain. And so that whole hollow area, and then our subarachnoid space is filled with this cushioning nutritive fluid called cerebrospinal fluid. We've got protection from foreign matter that does manage to sneak its way into this general space. So our microglia are doing phagocytosis, eating up foreign matter, foreign particles. And we've got two different types of cells that form myelin. We talked about myelin, helpful for speeding up conduction of action potentials. We've also got a zoomed in picture with those different types of cells labeled on, those different glial cells. Okay. So the reason we do want to make sure that we are aware of the glial cells is that they do have important functions to play, and when they get damaged, it can lead to disease. Specifically, can lead to neurodegenerative diseases. So, for example, MS, multiple sclerosis, has to do with a loss of myelin, which means you end up getting kind of like blurry conduction of action potentials and nervous system function more generally, which leads to lack of control of different parts of your body potentially from that loss of myelin, right? So we know that oligodendrocytes and the Schwann cells are responsible for making that, right? That myelin's important. Alzheimer's disease is related to loss of a certain type of neuron called a cholinergic neuron, but they also get replaced by scar tissue and plaques, right? So that shouldn't be happening. Sometimes we can have almost an overactive immune system working in the brain. Parkinson's disease also is linked to a loss of neurons, specifically in this case, dopaminergic neurons, so neurons that produce dopamine. But our glial cells in Parkinson's are also, again, kind of misfunctioning, producing extra inflammation that unfortunately has the effect of damaging the brain a little bit further. Yeah. So a quick example type of question. Coming up. Why is that not coming up? So an example type of question on this material level that you should know it is basically being able to recognize the basic types of glial cells. So I might ask you, which of the glial cells is involved in producing cerebrospinal fluid? And you would say, Great. So those would be the ependymal cells. So this is the kind of thing you might put on a flashcard, basically. All right. So some just general other things we want to know that we're familiar with as we think about the central nervous system, because we'll be referencing these later on. So we want to know some stuff about the general shape and structure of the central nervous system. So we've talked about a fair amount of this in lab, I suspect. So this hopefully overlaps with stuff you've already done. But here we're looking at a picture of the main parts of the central nervous system. So brain, spinal cord, we're thinking. So we know that we have protection for the central nervous system in the form of the skull and the meninges that surround actually both the brain and the spinal cord. Cord itself being protected by vertebrae rather than the skull, but both have meninges. Okay. So that's physical support in terms of not getting too knocked around. But we also have our cerebrospinal fluid that you may or may not have thought all that much about as you were touching the bodies, because most of it drained out when we pulled out the brains. But this is important for maintaining function of the brain, this watery kind of clear bath. Does some physical cushioning, but also helps supply the brain with what it needs. So that's coming from those ependymal cells, specifically in a region called the choroid plexus. So a region lining kind of the outside of the ventricle. That CSF, that cerebrospinal fluid, does flow through a path, right? So it's being produced in the ventricles. So here we can see lateral ventricles. It flows, moving through these little gaps. Right, so here we can see it moving into the third ventricle, located kind of centrally in the brain. And it can flow down through the cerebral aqueduct to our fourth ventricle. So from there, some of it's going to go down towards the spinal cord, and some of it's going to escape out and go in our subarachnoid space and surround the brain overall. Eventually it gets reabsorbed and ends up basically in your veins, returning as part of that fluid towards the heart. So you may have noticed like some really rough parts on the dura. So those are little pieces of evidence of where we had projections of the arachnoid layer of the meninges projecting towards your dural venous sinuses and kind of letting you let off some of the pressure of that cerebrospinal fluid when necessary. Okay. So from a physiological perspective, this is also maintaining a stable interstitial fluid environment. So when we say stable interstitial fluid environment, you mean like we've got the right concentrations of ions, we've got things that we expect to be dissolved in certain amounts in cerebrospinal fluid. So here we see a comparison of what some of those things are in your plasma, so the extracellular fluid in your bloodstream, versus what it looks like in the cerebrospinal fluid. So we can see that they both have glucose, proteins, and a variety of ions, but there are differences between those concentrations. So this is why you don't want your whole brain just like floating in blood, right? Is because you've got different amounts of these different components, right? You don't want that much protein in the fluid around your brain in particular. Right? You can see there's way more protein in plasma than there is in cerebrospinal fluid. You do want a good amount of glucose. You do want a good amount of these ions, which hopefully makes sense because now we know that neurons function based on the electrochemical driving forces that we partially set up by setting these different concentrations of ions. So we're also going to mention blood. So this is going to be important when we think about how our endocrine system is going to prioritize where it sends glucose under different conditions. So that's why we're mentioning it now. So this will kind of play in more later. Okay. But you want to know that while your central nervous system is relatively small compared to your body weight, your body size. It's only like 3 or 4 pounds, about 2% of your body weight. It gets a disproportionate amount of blood supply. So it's 2%-ish of body weight, but 15% of your blood is going there, right? So your brain is really important for functioning of your body. We get lots of blood going to the brain to supply oxygen, to take away carbon dioxide, and to make sure that we have glucose so that we can do metabolism, make ATP, so that we can power those sodium potassium pumps, which we know are helping set our resting membrane potential, for example, so that we have something we can shift to send action potentials, to send signals. Right? So we need good blood supply to the brain for it to function. Yeah. The brain has a particularly high metabolic rate, so meaning it uses a lot of energy. Right? So it uses a lot of energy, makes a lot of energy. So your brain is getting 15% of your blood. It's using 20% of your oxygen, right? It's really greedy for these life-giving molecules, right? So it's using 20% of the oxygen, and it's using of the glucose, right? So this is why carbs are actually pretty important for you, right? Like when you are at rest, half of the glucose in your body, so half of that super easily available sugar, the main one that we trace down when we look at how do we make ATP, half of it is going to your brain unless you're doing something active, right? So we need lots of blood flow to the brain. So it's got energy, it needs oxygen to break down that glucose. We need that blood going there. So this is partially because we need to continue doing metabolism in the brain. So our neurons are going to be dependent on aerobic metabolism. So they do glycolysis and they do the rest of the steps, but they're going to need oxygen. So they need all that blood coming there. They need fresh blood. They need to get the blood kind of immediately before anything else takes oxygen out of it. So when we look at blood vessels coming out of our heart, we're going to see a lot of them are going boop, straight up towards the brain. Okay. So part of why they need so much food is that they don't store energy really until later. So you'll have noticed that the brain didn't really have a bunch of fat globules all over it, which you will see in other parts of the body. I'll try to remove them for you. So we don't have a lot of fat up there. We don't have glycogen, which is that storage molecule for glucose where we have a bunch of them packed together. There's a little bit in glia, but definitely not in the neurons. Okay. We don't really use fatty acids for energy. So we talked about the fact that we can break down things other than glucose for energy, but not so much in the brain. Right? The brain doesn't have a lot of the machinery for that. In extreme conditions, or if you are on the keto diet, it can use something that's not glucose. Specifically, it can use ketones, right? So we've got one alternate option in terms of fueling the brain, but that's kind of it, right? So we need all this blood flow. We need these things immediately. We don't have as much kind of diversity in what kinds of molecules we can break down and use for energy. We don't have as many options for ways we can make energy. Yeah. So it's a problem. You don't get blood supply to the brain. Right? That can happen if your heart isn't pumping, right? You have a heart attack, right? That causes a problem, but more common when we're thinking about issues that are caused by brain blood supply, it's a stroke. Right? So a stroke really is referring to a decrease in blood supply to the brain, usually because we've blocked a blood vessel, right? So when you have a stroke, what usually happens is you got a clot in your blood somewhere, usually somewhere else, travels through the bloodstream and gets stuck in one of those teeny tiny blood vessels that we've seen in lab. And when you get a clot stuck in a teeny tiny blood vessel, blood doesn't flow anymore through that vessel, doesn't get to the regions of the brain that should be supplied by that blood vessel. That's part of why the circle of Willis being a circle. is kind of important because that gives you some alternate routes for blood flow to get around blood vessel damage, but that only works so well. So occlusion here means blockage. The other thing that can happen is, bleeding. So hemorrhage would be when you have some type of damage to a blood vessel, and then you're not going to get blood to the parts of the brain that are past that because you're losing it out that injury. So an example of this that isn't like a car crash or trauma, which is like an aneurysm. When an aneurysm bursts, that's like a swelling in your blood vessel that then pops under too much pressure, right? This is a way you could end up with like hemorrhage in your brain. So we mentioned the blood-brain barrier when we were talking about astrocytes. So let's take a look at that because we're going to be talking later on extensively about capillaries and how they work everywhere else. So we want to mention that they work a little bit differently in the brain. Okay. So usually what a capillary is a teeny tiny blood vessel made basically of a layer of endothelial cells. Okay. So we're going to see that capillaries are where you exchange stuff between the blood and extracellular fluid that surrounds the rest of your cells. Okay. So that exchange happens partially by going through the cell, so we can transport things across one side of an endothelial cell and then through the other side of the endothelial cell. And if our endothelial cells are packed close together and have tight junctions between them, a tight seal, then we really have to go through a cell. We can't go between any gaps. But we're going to see that in some of our capillaries later on, get a little bit leaky, right? Varying degrees depending on where we are. Right? So capillaries in your kidney are going to be specialized versus capillaries out in other parts of your body. But what the brain capillaries have is this specialization to make sure that those endothelial cells are really right next to each other, packed tight, closed, absolutely nothing leaking between the gaps, right? So we've got these tight junctions sealing our endothelial cells together. We've got these astrocytes kind of holding on to the endothelial cells, helping us create this barrier between the blood and the cerebrospinal fluid, or extracellular fluid here, that helps us make sure that anything that gets into the brain or gets out of the brain is being at least processed by one of these endothelial cells first. Right, so we can block things like proteins, we can block all kinds of stuff that might otherwise sneak between our endothelial cells of the capillary. Let's also take a quick dive looking at the myelin specifically, because we had two types of glial cells that were responsible for myelin. So we're going to have our oligodendrocytes and we're going to have our Schwann cells. So we know what myelin does for a neuron. Right? We create a coating, basically an insulation, speeds up propagation of an action potential because we have that saltatory conduction jumping from node of Ranvier to node of Ranvier. But if we were to specify whether our neuron is in the central nervous system versus our peripheral nervous system, we see slight differences in where that myelin is coming from. So here we're looking at what myelin looks like in the peripheral nervous system, which if you see a picture of a myelin sheath, it's probably showing you a picture of a peripheral nervous system nerve. Okay. So here we see that structure around these nerves in the peripheral nervous system got almost this kind of bead look to it, right? So we can see that we have a cell that wraps around and around and around and around the axon in these little segments. Okay, so each of these is made by a Schwann cell, okay, wrapping around. And we call these structures that they're making here the neurolimb. So that's what we're looking at when we look at our peripheral nervous system. So this is what we were drawing mostly when we were talking about how the conduction is different in a myelinated neuron. Here we can see an actual histological slide. So here you see the Schwann cell's cytoplasm here around the outside, and here you see how it's wrapped lots and lots of myelin around the axon of a neuron. You can see some more Schwann cells over here. Okay. So when we're thinking about which neurons get myelin, which ones don't, it's usually small axons, small neurons that are going to be unmyelinated, and it's going to be bigger ones usually that have myelin, right? Because we know that large neurons, large diameter, that's another thing that's basically an adaptation for fast movement of an action potential, right? So if we're trying to make a nerve work fast, helpful to make it big, helpful to put myelin on it. Okay. Even when you have small neurons in the peripheral nervous system, there will be a little bit of a neurolemma around it. So like a little bit of a coating, but not this big multiple wrappings of the Schwann cells making big myelin chunks. Let's compare that to the central nervous system. So when we're looking at that arrangement with the Schwann cells, that's like out in your arm, let's say. So now we're thinking about what does myelin look like? We're thinking about the brain. So we still have a situation where we have insulation around the axon of our neuron, but you can see that the cell making that myelin looks different, right? Instead of being just one little bead around one axon, you can see we've got this kind of octopus looking thing, right? So that is our oligodendrocyte. You can see it's reaching out to hold on to multiple axons, right? So it's not dedicated to just one neuron, not dedicated to just one patch along the axon of one neuron, doing a lot of jobs kind of at once, okay? So one oligodendrocyte is going to help out multiple neurons versus a Schwann cell is going to be dedicated to just one peripheral nervous system neuron. Okay. So the structure is different, so we wouldn't call this a neurolemma. We wouldn't have that multiple wrappings made by one cell like we do in the peripheral nervous system, but we do speed up conduction a little along the central nervous system neurons. And we do have a change in color when we look at these myelinated axons. Okay? So if you think about the brain, you mentioned gray matter and white matter, either in lab or elsewhere in your lives, what you're actually noticing when you see parts of the brain that are different color, right, you're noticing that myelin, right? So you might have noticed that, for example, like in the corpus callosum, it had a different color, right? That's because of this myelin, right? So that white color, that white matter, has to do with these farther kind of connecting neurons here. And when you see tissue that's grayish, like most of the brain, when you kind of held them and looked from the outside, that grayish pinkish color, that's the cell body of a neuron and dendrite, right? So that's the part of the neuron that doesn't have myelin wrapped around it. So I mean, another slide for you guys is basically our other form of kind of cheat sheet for learning these different glial cells. You want to make sure that you can tell apart our myelin in the central nervous system versus what our myelin in the peripheral nervous system looks like. And remember that one sentence version from before, but here we have kind of illustrated out again those differences between the two. Any questions at this point? We got a little time for review, so I need some review activities for you to work on and to kind of spur some questions. We can grab them from the front. Counting papers when we pass them back, running out of it. So what these are walking you through mostly is how we look at the different parts of our action potential, how we look at the different parts of our synapse and so on. And I can send you guys the link for this as well, but here's what we're walking through here. So we're taking our graph of the action potential along with our permeability of sodium and potassium. We're making sure we can fully describe what's happening here at A, what's happening between B and C, what's happening between C and D, and so on. Okay. And then you're going to make sure that you know the parts of the neuron. So you're going to draw me one and label it. We're going to do some synapse labeling. And then we have a little bit of practice here on those modulating neurons and those postsynaptic potentials that we've got. Okay. All right. So I'll post that link, or you can grab a paper copy or both. And there's a link to the key at the bottom.